Hey everyone, good morning. It's my pleasure to tell you a little bit about Opus Genetics, where we're developing gene therapies for inherited blindness. So it should come as no surprise that there are millions around the world affected with inherited retinal disease. You might even know someone who's affected, someone who has visual impairment, or might even be legally blind. The good news is that we know that we can use AAV viruses to replace broken genes. This is a gross oversimplification, but it's really just to make the point that right now AAV vectors are the tried and true way to get gene transfer in the retina. And we need that because this is a really big, hairy problem. If you've seen this bubble chart before, you know there are over 280 genes are known to cause monogenic blindness. Now, some of these are easier to go after than others. Some fit inside an AAV, some are too big. There's syndromic disease, there's recessive, there's dominant. And some progress faster or slower than others. But our view is that there's a lot of low-hanging fruit we can go after with the current toolkit that we have right now. And in fact, our motto at Opus Genetics is to avoid risk on risk. We only work with AAV capsids that have proven safety and efficacy and manufacturability. We only deliver by subretinal injection, because that's the tried and true way to get good gene transfer in the retina without inducing a lot of inflammation. And then manufacturing is not a scale-up problem like you hear about in other therapeutic areas for gene therapy. This is boutique, small batch manufacturing. We just need really high-quality material. So our first program here is for LCA5. This is a rare, severe pediatric blinding disease. In fact, these kids are diagnosed in their first year of life, sometimes in the first few months, because they can't even fixate. So their eyes wiggle with the nystagmus, and the parents can tell there's something wrong pretty quickly. The good thing is that even though they have terrible visual function early on, their structure remains for decades. So we call this structure-function dissociation. Probably one of the most important concepts in retinal gene therapy, because if you have a way to restore function, you need to have the tissue there that's viable for rescue. And in fact, Gene Bennett and team at Penn found that with the knockout mouse model, as long as there's at least 30% of the ONL remaining, you can rescue structure and function. So we're happy to be in the clinic with this first program, phase 1/2 at University of Pennsylvania. We've fully enrolled our first cohort of all patients safely dosed. Safety looks great so far. But since it's an open label trial, I can tell you a little bit about some of the data that's coming out of the trial. But before I do, I want to give you the setup for the next slide. It's a video of an exploratory endpoint where we're using virtual reality that's a new version of the multi-lumens mobility test you may be familiar with, which is the primary endpoint used for the Luxturna approval. So what you're going to see is an avatar of a normally sighted person going through this challenge course where they're wearing the Oculus headset, they have two handsets, and they walk through the course following the arrows, tagging objects. As they tag the objects, they disappear so they know they tagged them. Some are big, some are small, some are low, some are high, some are moving, and then eventually they find the exit and complete the test. So imagine doing this at lower and lower levels of light as it gets harder and harder, so it's a real challenge for them to do. So let me walk you through a little bit of data that was just presented last month at the American Academy of Ophthalmology by our PI, Tomas Aleman. So these are two patients of data stacked on top of each other. The vertical axis is number of objects identified, and the horizontal axis is increasing luminance going from left to right. So the top left graph is control eye for the first subject. Baseline's in white, month 1 in orange. And basically you see they're on top of each other because this subject couldn't identify any objects, couldn't even do the test basically, just a flat line. The study eye at baseline on the graph on the right, you see the white circles. So again, couldn't identify any objects at baseline, but then 30 days after treatment with a low dose, big increase in ability to do this test, identify objects. At higher and higher light levels, you see more and more objects identified. So pretty significant improvement in functional vision. Second patient, control on the left. Their control eye, actually, they could see fairly well their control eye. Baseline month 1, pretty much on top of each other. Study eye on the right, bottom. You see that at baseline, only at the highest level of luminance could they identify one object. And then after treatment, at the second lowest level of luminance, they could identify lots of objects. So again, whopping response, Early data, preliminary endpoint, but super excited to see what we're finding so far in this very severe IRD. So a little bit about our second program. This is called BEST1. This is a severe central maculopathy. It's what we call a top 10 IRD. It's a fairly prevalent IRD. It's over 13,000 patients, US, UK, Europe combined. And it involves a broken chloride channel that leads to these characteristic egg yolk-like lesions in the dominant form on the left, and scrambled eggs is the sort of the recessive form on the right. Both are bad. Both can cause catastrophic loss of central vision. But we're excited to get this gene therapy into the clinic in the first half of next year. So I want to end with a little bit about the commercial picture here. So when you think about rare disease, you think about, oh, well, there's not very many patients. If you follow this space, you know that Luxturna, the first one approved in 2017, it's actually the least expensive gene therapy on the market. It's $850,000 per person. When we do payer studies, we find that that's definitely the floor. There is price elasticity here in this space. So the simple way to think about it, at least in the US, is for every 100 patients that are dosed, that's $90 to $100 million in revenue. So if you think you've got 1,000 patients, that's about $1 billion. 10,000 patients, et cetera. So pretty easy math. We actually think if you have a stack of assets in this space, it's a really compelling commercial story. And finally, I'll just leave you with this mosaic of these patients. They know who we are. We know who some of them are. They actually allow us to use their names and likeness to create awareness. But that's why we're doing it. We're here to help them see the world around them. And thank you for your attention.